Patrick. Patrick, Claire, and all at Dassault Systèmes. It's a great privilege and my pleasure to be here this morning with you. And I'm going to try to share in the next 25 minutes how the field of oncology is changing through the advent of new systemic therapies and very specifically T-cell engagers. So, engineering immune synapses is forcing the immune system to create artificial synapses to bring in close proximity T cells with cancer cells. This has been achieved for hematologic malignancies with success over the last 10 years and has proven much harder for solid tumors. This is my disclosure. It's pretty obvious. I work at Amgen. I'm paid by Amgen, and therefore my view might be related to that reality and safe harbor statement. I'm forced to do this. But this is the topic I want to deal with you. I'm going to review what's a T-cell engager, what is the potential of this modality, where does it sit in the overall anti-cancer possibilities of systemic therapies. Then we're going to see why it has taken more than 10 years to make T-cell engagers successful in solid tumors, what are the drivers or the challenges of that. And then I want to try to give two very concrete examples of T-cell engagers in solid tumors that have either registered status or ongoing in phase 3, and then wrap this up by some conclusions on how this will finally play in the world of cancer. So, cancer is still treated by three main pillars: surgery, radiation, and systemic therapies. One thing I want all of you to understand is that these three pillars are communicating more and more each other. If you think about radiation therapy and systemic therapy, well, you have a completely new modality that is radioligand therapy that has emerged, bringing the power of the atom to targeting a specific tissue or cell. And I'm pretty sure in the upcoming decade, we're going to see an explosion of the conversion of surgery with systemic therapies, because, you know, a surgeon, once he opens up and is there, I don't see why he could not use a polymer gel that will allow systemic diffusion of smart therapy. So I'm sure the conversion of these three pillars is going to become more and more frequent. Now, going back to the systemic therapies that are the core of what oncologists do, well, they have allowed us, along with surgery and radiation therapy, to improve the survival of cancer patients. And this picture captures it nicely. It shows that we have moved from having around half of the patients diagnosed with cancer alive at 5 years to 2/3 of them today. That's still not good enough. A third of patients dying. The way we improve the outcomes varies by tumor types, but this is the sense of urgency we need to have, correct? Cancer is one of those diagnoses that are very brutal for people who experience it, and they're brutal for the entire family. So we need to do better, and we are lucky in the advancement of, I would say, biology and biotechnology that has allowed the diversity of systemic therapies to be enriched. The color of my hair tells you that I'm old enough that when I was a fellow treating cancer patients in the mid-90s, basically the only thing we had was hormonal therapy and chemotherapy. And you know, you were permutating chemotherapies, but since then, in the early 2000s, we had the advent of molecular targeted agents, oncogene de-addiction. Then in the 2010s, the advent of immune checkpoint blockers, cell therapy, ADCs, and T-cell engagers, which are close to the immunotherapy and cell therapy, but have a different flavor. And that's the focus of what I want to share with you today. So I'm going to go in the science of T-cell engagers, but since we have a diverse audience, I will try to convince you not only through science, but also through revenue that T-cell engagers are changing the landscape of oncology. This is not me. This is Evaluate Pharma making projections about how T-cell engagers are growing as a market. People would have said that 5 years ago for ADCs, and most people were, we don't believe on that. Well, you see how big it has become, the wave of ADCs. T-cell engagers are growing because they harness the power of immunotherapy in a more precise manner, in a more targeted manner, and are unlocking some of the immune checkpoint blocker PD-1 insensitive tumors, as I will show you. So again, what's a T-cell engager? Well, to make things simple or simplistic, it's a bispecific antibody. Usually, you can be more complicated and have trispecifics, but basically it's a bispecific antibody where with one arm, you grab the T-cell through the CD3, and with the other arm, you grab an antigen from a cancer cell and force them to come in close proximity, therefore activating the T-cell to kill the cancer cell. And it's very powerful. And because it's very powerful, that's why we have had so many problems in trying to develop them in solid tumors, because the tumor burden usually is higher than in other settings like heme malignancies. On the right side of the slide, you can see how specific the T-cell engagers are. You have the synapses that are created when you have this in vitro experiment where the tumor cells express the antigen of interest for a T-cell engager. You have real synapses that happen, immunological synapses. But if you put in the mixture a T-cell engager that does not recognize any antigen on the tumor cell, nothing happens. They just stay around. So it looks fantastic, but in fact, it has been a very arduous and painful journey. And I assume one of the reasons Amgen hired me is to help them achieve and accelerate this development, correct? So what has been hindering the development of T-cell engagers? First, target selection. For heme malignancies, you can use lineage-specific targets that are very clean, and you can survive without CD19s and CD20s. You have problems with infectious diseases and others. But for solid tumors, if you don't pick a very clean target, it does not go well. It's too powerful, you have too much toxicity. And because you have so much toxicity, you have two phenomenons that most clinicians were not used to it, which is cytokine release syndrome and ICANS, which are neurological problems that can happen when you have an overactivation of these T-cell engagers in the brain that make, therefore, the phase 1 development of these agents protracted, difficult. You need to do a lot of step dosing. When you create bispecific T-cell engagers, sometimes you're obsessed by what you want to do, but then you don't pay attention to these need to be produced in process development, and you create Frankenstein molecules, and you have a lot of ADAs, anti-drug antibodies, that have been a challenge in the field. The solid tumor microenvironment, it can be very difficult to modify. Pancreatic cancer is highly fibroblastic, et cetera, et cetera. And the time from the moment you put a T-cell engager in phase 1 to the time you declare proof of concept is longer than most other modalities, which has made many players in the field say, wow, complicated, complex, not sure we should do it. So let's talk about the target, which I told you is essential. Well, the problem is that most of the obvious T-cell engager targets were taken in the 90s and 2000s. As I told you, cell lineage-specific targets, hematological targets, and when you try to then penetrate the solid tumor space, you have no more the low-hanging targets, but you have difficult ones to grab, where it's difficult sometimes to create the right antibody for the right antigen, and the antigen needs to be clean, very cleanly expressed in the tumor cells and not in healthy tissues. So clean target is one of the essential elements of success for T-cell engager development. I will not have time to go into it, but we as others embark on targets we found nice and appealing because were expressing many solid tumor targets. Monoclonal antibodies had shown some activity. And then we went with T-cell engagers, and we had a lot of toxicity because it's such a powerful synapse that you create that then you have a lot of side effects. And many T-cell engagers died that way. Then you also might want to go to a tumor where you have some degree of T-cell presence in the tumor. So you know that your tumor come in 3 flavors: those who are completely immune excluded, those who are hot, and those who are complete immune deserts. I think going in the complete immune deserts is more challenging than goes to those who are immune excluded, where the T cells are around the tumor. When you bring a T cell engager, at least the arm is around the city you want to penetrate, so it's a little easier. So that dimension was not completely taken into account. Then you need to have a good binding, and a binding that brings the right level of CD3 activation. And as I told you, you need to have the appropriate scaffold. So, I mean, Dassault system is about end-to-end everything, correct? Understanding everything. Well, here we probably overlooked how important it is to have the right attributes for a molecule to be developed. And the clinical development plan needs to leverage the learnings we have from the heme malignancies and bringing to the solid tumors, but also put some limits. When you have a T-cell engager and after 3 years you don't have a recommended phase 2 dose, I believe you don't have a therapeutic index. You might be in love with the molecule, but it means you don't have a therapeutic index, so you need to be pragmatic and say, I'm sorry, this is too complicated. I have not been able to overcome the complexity. So this is a slide that I like because it shows you that you need persistence, and developing T-cell engagers is not for the faint of heart. So you can imagine if you were a pharma with only a 5-cycle tolerance for a modality, you will be out of this business very quickly. So Amgen acquired Micromet in 2012, by the way, a German company, Ariel, based in Munich. And they are the ones who invented this idea of T-cell engagers for heme malignancies in ALL. And we got that approved as Blinatumomab in 2014. It was approved in 2014, but today we are still getting new approvals for Blinatumomab in ALL because we learn, as I will show you in the next slide, that it's better to use it as early as possible in leukemia. We started backwards. And you see that then we started bringing the first T-cell engagers in the clinic for solid tumors in the mid-2010s, and we got IMDELLTRA, which is a DLL3 T-cell engager, approved last year in May. So it took a long time. And now we have more assets, and I will give you some details on what's IMDELLTRA and xaluritamig. So Blinatumomab is the prototype initial T-cell engager. It was a canonical BiTE. In Amgen, we call it BiTE because it's bispecific T-cell engagers. It's a trade name we have, so we call it BiTE, but the field calls T-cell engagers. And look at how Blinatumomab was developed. It was first positioned in the worst class of patients, patients who were refractory to chemotherapy in ALL, and then we moved these to patients who were minimal residual disease positive, and then we moved it recently to the first-line setting in consolidation, and now we have ongoing phase 3 trials in induction or chemosparing, and we converted recently the Blinatumomab use of continuous IV to sub-Q, which is going to be friendlier for patients and friendly for the health systems. But it took a long journey, but a lot of lessons we learned through the use of Blinatumomab in heme for solid tumors, and I would show you how we apply it for the development in small cell lung cancer. So when I saw that, and when I joined, I said, wow. Here we need to avoid this 10-year development plan. It's not going to go well for me. I don't think I'm going to survive 10 years of development of a T-cell engager in solid tumors. So let's make sure we bring a T-cell engager in solid tumors to the earlier disease setting as quick as possible. And the tumor burden clearly is something that is related to the toxicity. So if you try to bring the T-cell engager to lower tumor burden, you're going to do better. So let me move to the two T-cell engagers that I would like to give you some flavor. The first one is half-life extended BiTE called tarlatamab. IMDELLTRA now is commercially available in the US, and it's developed for small cell lung cancer and neuroendocrine tumors. The approval is only small cell lung cancer. And the second T-cell engager I will share with you is for prostate cancer, notoriously cold tumor. Immunology has not cracked prostate cancer. PD-1, CTLA-4s, CAR-Ts have failed in prostate cancer, and we have really encouraging results, encouraging enough for us to have 2 ongoing phase 3 trials. And what was the unlocking element of that is the genius of the 2+1 approach. So this is a T-cell engager that, if you want, binds twice to STIP1, because STIP1 is expressed to some degree in healthy tissues, and to avoid the toxicity, we created this 2+1 approach, and I will tell you more in one slide. So IMDELLTRA leveraged a target that we didn't discover called DLL3, which is exquisitely expressed at the surface of cancer cells of neuroendocrine origin. We're very lucky because DLL3 in normal tissue is also present, but never at the surface of the cell. It's inside the cytoplasm, okay? And that gave us a fantastic target for TCE. Many pharmas were like, I don't want to touch DLL3 because there is a biotech called Stemcentrx that developed a DLL3 ADC that was acquired by a big pharma, and then it failed in phase 3 in a very expensive development plan. I did one of the phase 1s of that asset. I knew it was powerful, I knew it was active, and I knew it was toxic. But you know, once you have highly skilled people doing phase 1 and then you put it in the hands of everyone, if the toxicity was not fine-tuned, that's how it failed. But we went for this target with a different modality, not from an ADC, but from a T-cell engager, and we saw activity in phase 1. And when we saw activity in phase 1, we decided to go bold and immediate. So it was not common to have something showing you response rates in phase 1 and then go and see the top of the house of Amgen and say, we want to launch 4 phase 3 trials on the basis of our phase 1. But that was the lesson from Blincyto. I cannot wait for one to be positive, do the next one, do the next one. I will be here coming back in 10 years with completely white hair. So we went against all these indications, first to confirm the results in second-line small cell lung cancer, to bring it in the front line of small cell lung cancer post-chemotherapy, that's the maintenance trial, then to bring it to the very front line in induction maintenance. And then we also say, well, in small cell lung cancer, there are people who do not have metastatic disease, who are treated by chemoradiotherapy and still relapse in more than a third of the cases, way more. Let's bring Blincyto there because the tumor has already been shrunk by chemoradiotherapy, so we launched also a limited stage consolidation phase 3 trial. So we randomized tarlatamab against the standard of care that was the different chemotherapies that were used in the different parts of the world. And we made an overall survival endpoint. And here I want to share one of the tensions we have today in oncology, including discussing with the regulatory bodies, which is OS and crossover, okay? So if the rules of engagement of FDA are, we want an overall survival trial, the rules of engagement for me is to say, I'm not going to do crossover. And people say, well, that's not ethical. Well, I say, if it's an experimental agent and FDA wants me to show an overall survival benefit, it means it's experimental. If we are convinced everyone should get it, it should not be experimental. So we didn't do crossover because we wanted to show a real overall survival benefit. And this is exactly what happened. And it's very rare that you replicate in phase 3 the results of a phase 1. So we improved the median overall survival very largely. You can see that the separations of the curves is very clear. We went from 8.3 months to 13.6 months. 13.6 months of median overall survival in small cell lung cancer in second line is better than the median overall survival with standard of care today in first line. This is how good the data was. And that was great. We also showed that objective response rate were higher, 35% versus 20%. Our phase 1 showed 40% response rate. So to have something as close as that was very good. And not only we have more responses, but they last longer, which is great. And is one of the characteristics of immune modality, that power to sustain the responses. So that's the story of IMDELLTRA that is still ongoing. And we are very excited by what this will do in patients in the frontline setting and beyond. I was very happy at ESMO in Berlin because we presented some of our phase 1b data of IMDELLTRA with the use of it in the frontline setting with the combination of PD-L1s and chemo. And the discussion conclusion was, wow, wow, which to me was, a great hope for the patients and shows the power of this modality. Because in fact, small cell lung cancer is a very aggressive disease. It's one of those King Kongs that proliferates very quickly. And it's great to see new therapies help patients. So let me tell you the story of STIP1 in the last 3 minutes I have. STIP1 is a target that is expressed in prostate, and for which, as you can see, we also went very broad in development. And the genius was the collaboration we had with Xencor, which was convinced that manipulating affinity, meaning how much you bind the target, can influence the selectivity of your T-cell engager. So a lot of people didn't want to go to STIP1 because they say, well, Soria has been telling us we need to have a clean target. STIP1 is not so clean because it's expressed at high levels in cancer cells in the prostate, but it's also expressed at low levels in healthy cells. So how do you deal with that? Well, you deal with that with a T-cell engager that is like a monkey that tries to grab twice to STIP1 if you want. If you go to healthy cells, there are a few STIP1. So one arm of the monkey can grab a STIP1, but the other STIP1 is so far away that the other arm of the monkey is not going to grab it. Therefore, the tail of the monkey doesn't activate the T-cell engager. If you go to the cancer cell, there is enough of STIP1 that you grab both, then the tail can bring the T-cell, and that creates your therapeutic index. That's the beauty of of manipulating and improving the therapeutic index just by how the power of biotechnology can increase. This is the waterfall plot we saw in terms of PSA reductions, very, very promising, but more important than PSA drops, where objective response rates in prostate cancer, shrinkage of tumors in the liver is very difficult to achieve. The liver is an extremely immune-excluded environment, immune-cold environment. So to see prostate cancer, liver metastasis shrinking is great. You see that PSMA PET that shows activity, very exciting. We have now 2 ongoing phase 3 trials, and our vision is the same. We want to bring this asset earlier, not only in hormone refractory metastatic prostate cancer, but also in hormone-sensitive metastatic prostate cancer, and earlier if we can. So to wrap up, these are 2 eyeballs showing you on the left how many T-cell engagers are being developed in hematological malignancies. What you want to be is in the green of the eye. That means you got approval. So we were the first with CD19 Blincyto, but you see many others have been approved. In the solid tumor space, there are only 2 approved, but many others being developed. And today, we have 9 T-cell engagers that have received FDA approval, 7 of them in hematological malignancies, 2 of them in solid tumors. The first was by Immunocore in uveal melanoma, but it's a peculiar T-cell engager because it needs, it's HLA restricted. So therefore, it's more narrower. We were the first to get an approval of a T-cell engager without HLA restriction in a common solid tumor, but these are great results. And to wrap up, I'm already over time by 36 seconds. TCEs are a new important systemic modality that is growing. And the fact that now they have shown activity in solid tumors is going to increase the investment of pharmas in this modality, very powerful. The clinical development needs to be a little bit like haute couture or three macaron restaurants. It cannot be McDonald's, it cannot be fast food. You need to understand what you are doing. You need to be very intentional. You need to use a step dosing in a smart manner and overcome the toxicities. We will face a problem with physicians not knowing what CRS is and ICANS is, so a lot of training of our colleagues in the community. This is something that can be handled. Do not worry. You just need to understand what it is. And creative engineering will change the world for T-cell engagers. There are so many fantastic new modalities to create them, trispecifics to deal with T-cell exhaustion, dual targeting. Now at Amgen, we are developing square bites, so that target 2 different antigens to create a white space. So I hope I share with you my enthusiasm, and I thank you again for your attention.
Thank you very much, Professor. We are very honored and humbled by your presentation. I will immediately open to questions for the room. I suspect there are many. Any questions from the room? If not, I will start.
Of course.
Let's start. You mentioned the haute couture for the clinical development, which I perfectly understand. I also understand your comment on the clinical trial process. So are you also intending with those TCEs, new approaches, to have a discussion with FDA and the regulatory body for them to adapt?
Yeah, it's an excellent point. And you can imagine I cannot discuss too much FDA interactions because we are recorded and it's complex. But it is obvious that the TCE modality is the new kid in the block, and therefore a lot of people are trying to learn with it. And we are extremely engaged with FDA about the tolerability profile, the feasibility, how do we bring this to the community, and we ourselves are spending a lot of time thinking, how do we help the community hospitals leverage and use this new modality, correct? Because small cell lung cancer, the sad story is a tobacco belt, so meaning, underserved patients where you really need to invest to make sure these communities have access to it.
Very interesting. And also another question that I have, you mentioned, of course, the awareness of the practitioner for CRS and ICANS. This really talks to us, at least at Dassault Systèmes and maybe data, because with Media.ai, we have so many projects leveraging big data lake for CRS and ICANS. So the question is, how big or what is the part of generative AI or data understanding in your process to leverage TCEs?
Yeah, that's a fascinating question. And one of the reasons I put together this deck is lessons learned, correct? If you have been doing this business for 15 years, you need to distill this with key lessons. And it was a painful process because everyone has his opinion.
Yeah.
The research people were, completely focused on certain criteria, the clinical people on these. The process development was like, good guys, bring us Frankenstein molecules. Can we talk before it's a Frankenstein molecule? So it was fantastic, and I agree with you. That can be done through human exchange. It can also be done by leveraging the data you have.
Yes.
And
not documents, really data.
Real data, and real data that needs to be in a format that is exploitable for AI. And it's one of the biggest challenges, especially when you go to the research lab book notes.
Yes.
You know that well. It's one of the frontiers that need to be unlocked. Same for the insights in the clinical development space. Same for the insights in process development, how you increase the productivity.
Exactly. And if there's one question over there or here.
Hi, it's coming. The mic is coming.
The mic is coming.
Hello, hi.
Hello.
Thank you so much for this presentation.
You.
Very exciting work. Quick question regarding the therapy you're developing. Are you considering, or did you consider the use of companion diagnostic to go alongside your drugs?
Yeah, so of course we always consider that, but DLL3 is expressed in more than 90% of small cell lung cancers, and in that setting we do not see a necessity. But we presented and published recently data in neuroendocrine prostate cancer, where the neuroendocrine feature is not embryological, is a consequence of therapeutic pressure, and in that setting you need DLL3. The responses we had in neuroendocrine prostate cancer were only in DLL3 positive neuroendocrine prostate cancer. But then you have the problem of who wants a biopsy in his prostate when it's advancing. I can tell you, man is not as courageous as women. They don't want anything getting close to the prostate. So we are trying to develop liquid biopsies approaches to deal with DLL3, but it's a great point, yes.
There was one question over there, Claire or Takeda.
You had a question? Sure? Okay. Hello.
Hello.
I'm Kathy from Takeda Pharmaceuticals. Just want to ask about, you got FDA approval, right? But looking back, now we have a new digital technology solutions available. Like, is that something we can do? I'm from the digital world, not scientists, right? What we can do to help you to accelerate the approval process. And then also, once you get approval, you're going to do the launching, right? How we can get a product to the patient faster. Is there something we can do to help you to accelerate?
Yeah, I think there are many things you can do. I mean, we have in our own company a very significant investment in real-world data, and that has helped us a lot in the negotiations to show regulatory bodies the consistency of the of survival in small cell lung cancer is terrible around the world. And we have the same survival in rich countries and poor countries. It means there are not very good therapies, correct? And we have leveraged that by showing how consistent is the survival in Albania, France, Bolivia, US, once you are second or third line small cell lung cancer. That's powerful, and it cannot be done by the clinical trial. It has to be done by real world data. Yep. So there are many ways to accelerate.
Thank you, Professor, for this very inspiring presentation. I wanted to come back to the primary endpoints. How do you see them evolve in the field of cancer? And if you go to secondary, how do you impact quality of life for patients, and how would you measure this?
Thank you, Claire. That's a great question and a very difficult one, and one where we will need to navigate with what the regulatory bodies want us to achieve and the health economic bodies want us to achieve or not. Oncology in FDA had pioneered the possibility to get approvals earlier based on surrogate endpoints, like notably progression-free survival and others, MRD for hematological malignancies, multiple myeloma, ALL. But now they are changing a little bit that point of view, and there is a new draft that says OS it should be. Well, so the answer is the reality. We need to leverage all the data that we generate through patients who so generously participate in clinical trials with all the dimensions. And I think quality of life and patient-reported outcomes is just in its infancy, correct? And all these scales from ERTC and others are great, but they are from the previous century. We need to leverage digital data. I mean, it's just really fascinating to me that we don't use all the elements that an iPhone will capture in someone in phase 1 trial. How much you move, how much you are able to walk steps, etc., are indicators that you're doing better or not. So I think we are at the precipice of a completely new world of unlocking new elements that will justify the benefit.
Thank you very much. Well, let's thank again Jean-Charles Soria and give him a round of applause. Merci beaucoup. Merci beaucoup.